So step one in the digestion process of carbohydrates is chewing and mixing the saliva and the enzymes in that saliva with our amylose and amylopectin. However, the food does not stay in our mouth long enough to break these polysaccharides down into the monosaccharides. So that food's going to pass to the stomach. The stomach can further break down, let's say, sucrose. Sucrose is that disaccharide that has fructose and glucose. But we're going to just follow the track of our polysaccharides, therefore our alpha D glucose. The remaining saccharides are going to pass to the small intestine. There we have more enzymes that can finish breaking down and separating each of those monosaccharides, specifically our glucose molecules. In the small intestine, we have villi and then microvilli, so it's like little tiny hairs everywhere. Lots of surface area where these nutrients can interact with the intestine wall. When they get to their monosaccharide form, specifically focusing on our glucose and fructose, they then can pass through the microvilli into our blood capillaries that are constantly cycling through. Once it's in the blood, our body then needs to determine and regulate that glucose concentration. The pancreas is going to signal the beta cells to release insulin. From there, our pathways can go one of three ways. If our blood glucose levels are high, the insulin is going to go to the liver cells, telling the liver to convert and form glucose into glycogen. If we need this energy, it can go into tissue cells and be used for cell respiration, and it can go into muscle cells. And if we have enough glucose, then the muscle cells are also going to convert the glucose into glycogen for storage. This is why an athlete can run longer or work out longer, push their body harder than we might think is possible, because we have these glycogen stores in the muscle to start breaking down and having glucose ready for cell respiration as needed. If we are maxed in both our liver and our muscles, it can go to our adipose cells. So our adipose tissue is our fat tissue. And in order to be in that tissue, it's going to get converted into fatty acids that then can be stored in our adipose tissue. So we have different ways in which we can shuttle this now absorbed glucose. Now that we have gotten our monosaccharides into the cell, we are going to follow specifically glucose through the remaining process of catabolism, finishing up with what we call cell respiration. The flow chart we're going to be going through, you have a copy of in Canvas, so you can print it off and fill it out. You can put it into your tablet if you're going to fill it out that way, or you can just draw this flow chart in your notes. Following glucose first. I find as we go through glycolysis and then into the citric acid cycle, tracking the number of carbons helps us make sure that we are accounting for all of the original reactants in this process. Glucose, once it's in the cell, is going to undergo glycolysis, which is a 10 enzyme catalyzed series of reactions. This is a linear metabolic pathway. Now, glucose at this point is typically going to be in the Haworth structure, so it does need to open up into the Fischer projection because that is what is going to be reactive. And it also needs to become reactive because it is relatively stable. So we've got to kickstart this reaction. We've got to put energy into it so we can get to that activation energy. Therefore, we are going to use 2-ATP. ATP is adenosine triphosphate. This is our chemical energy molecule within our body that runs a lot of our biochemical processes. When we cleave one of those phosphates, that is what provides energy for changing a structure or promoting and pushing a reaction forward. So 1 phosphate per ATP is going to be tagged or added onto our glucose. And this is going to be a series of reactions, it's just not one. But ADP, adenosine diphosphate, is that resulting product. Ultimately, we are going to get fructose, so we have some rearrangement of our atoms to make this isomer. 1,6 bisphosphate. Bis is a common term for 2. We really are moving away from the use of bis, but it's still in this name. So each phosphate from the ATP got tagged on to our fructose. The purpose behind our phosphate is it will increase the instability of this organic compound. That means it increases the reactivity of the compound. So I think of it as like a tag for enzymes to know that this compound is ready to undergo further reactions. That's how I think of our phosphates. Now, fructose can then be broken down, and this part is called the lysis or glycolysis, splitting up our glucose into 2 3-carbon compounds. One product of this lysis is glyceraldehyde 3-phosphate. We have made it full circle essentially at this point because the compound that comes out of photosynthesis is glyceraldehyde 3-phosphate. That is the backbone that we use to make our carbohydrates, our proteins or amino acids, and our lipids. Therefore, at this point, if we need to go make amino acids or go make fatty acids, we have this base structure just like plants do. So cool. Now, depending on where we are going to go from here, G3P can go in one of 2 directions. If we're kind of stagnant and we can't really move forward with cell respiration, G3P can be rearranged into another 3-carbon compound. We call this DHA or dihydroxyphosphate, another 3-carbon compound. The enzyme or the pathway that this is called is the triose, so 3-carbon sugar phosphate, because there's a phosphate on it, isomerase, or the formation of isomers of these 2 compounds. DHA is really important when we talk about fatty acid metabolism and amino acid metabolism because this is the 3-carbon compound made when those macromolecules are broken down. Then DHA will then convert over into G3P to continue through the process of glycolysis. DHA will also just be a storage molecule if we do not need G3P to move forward and create ATP as well. So we've got this nice little cross-section that will occur between these 3. We are going to follow through with our G3P. So we're going to take that and see where it goes through the rest of glycolysis. So G3P or glyceraldehyde 3-phosphate. It is going to undergo some conversions to make another 3-carbon compound that is specifically called pyruvate. Pyruvate is pyruvate is the product of glycolysis. It is the reason why glycolysis exists because then pyruvate will move forward into the mitochondria. Now, if you look at the structures between our G3P and our pyruvate, you can see that we lost our phosphate. That means that we are able to generate some energy. We actually get a little bit more energy out of this process because remember there's 10 total steps and I'm showing it to you in 2. But 4 ADP will ultimately make 4 ATP. We get a net of 2 ATP through the process of glycolysis because remember 2 were needed to kickstart this process. 4 are made. So 4 minus 2 is 2. Glycolysis is not an energy producer. It creates just enough ATP to keep itself self-sustaining. So we lost that phosphate. If you also notice that we have created a ketone and a carboxylic acid in this process. As we know, when carbons increase their carbon-oxygen bonds, that carbon is becoming oxidized. Therefore, there are 2 carbons in here that are undergoing oxidation. An aldehyde to a carboxylic acid and a secondary alcohol to a ketone. So if these are undergoing oxidation, we need a substance that's going to be undergoing reduction. The common compound that undergoes reduction is called NAD. It's NAD plus because it's going to be accepting electrons. Keep in mind, when we are looking for reduction, we're looking for an increase in carbon-hydrogen numbers. NAD is an organic compound. It's got some rings and some phosphates and stuff like that. You don't need to know the structure. But it's got double bonds it can break that we can add hydrogen onto. So it is an alkene hydrogenation process, essentially. We know all of this already. So cool. So it's going to be accepting those hydrogens from that glyceraldehyde 3-phosphate and its precursor. It's going to be accepting those hydrogens to become reduced. That product is NADH plus H plus. Don't worry about plus that H plus. That gets into a lot of technicalities. So now we made pyruvate. Through this process of oxidation reduction and phosphorylation, moving phosphorus around, we are so smart. From here, pyruvate can go in 2 directions. So at this point, the cell has to take into account, are we oxygenated? Do we have enough oxygen in our cells to move forward with what's called aerobic respiration or aerobic conditions? If it's going to move forward with aerobic conditions, the pyruvate is going to be shuttled and exported into the mitochondria. Remember, probably at some point in your education, you heard that the mitochondrium has been termed the powerhouse of the cell. This is where the majority of our ATP will be made. This is also where the other products of cell respiration, so the release of CO2 and water, are going to be formed as well. Now, if you are actively working out and you're in an anaerobic state, there's not enough oxygen to move pyruvate into the mitochondria and finish out cell respiration, we are going to undergo anaerobic conditions. So pyruvate can get rearranged into lactate or lactic acid. Looking at lactate, we still have that carboxylic acid, but notice that the ketone went back to its secondary alcohol. Therefore, from the process of pyruvate to lactate, that carbon got reduced or was hydrogenated. If it was reduced, then that means we need to have something that is oxidized. Well, we already have a compound in there that can undergo oxidation and reduction. It's its job to do this. So NADH that we have formed through glycolysis can give back the hydrogens and the electrons to perform that oxidation reduction process, reforming our NAD+. That can then go back up into glycolysis and help transfer our G3P to our pyruvate. With this, what we see in this first transition is we are going to see G3P get oxidized. which means NAD plus is reduced. We can then also see this process when pyruvate is reduced. And NADH is oxidized. So a lot of these processes are just oxidation reduction. Now this NADH, this is what we call a high energy electron carrier. Its whole job is to shuttle electrons. That is why it is there, to help move these bonds around in these organic compounds. Once we've built that lactate, it does get exported out to the blood. It will go to the liver, and from there it can undergo gluconeogenesis and then glyconeogenesis. It can be converted to glycerol to make fatty acids, or it can just make glucose to be reused when you return to an aerobic state.